how would you define epigenetics for somebody who's really coming across this for the first time? It's quite the buzzword these days, but I figure it's best to ask the expert.
Yes, so epi means on the top. And so on the top of our genes, there are molecular marks, molecular tags that can turn genes on or off, just like a dimmer switch modulates lights up and down in the room. And these are epigenetic marks. And epigenetic marks are so fundamental to the way we look and perform. For example, we have the same DNA, the same genes in every single cell of our body, and yet our eye cells look different from our hair cells. So the same information, the same hardware, but different software, different tags, epigenetic marks that program the genes to be turned on or off differentially in different cells. And this makes the magic of making these cells look and function differently from each other.
All right, so that's awesome, and thank you for making that absolutely crystal clear. Now, there's something in there that you said, which was epigenetic marks and just sort of methodologies that we can, or just sort of mechanisms for us to make those marks. And one of the ones that I increasingly come across and causes a lot of confusion is methylation. Do you mind just, and I know methylation is a very complex process, but if you're able to just sort of explain for us what methylation means.
Sure, and thank you for this question, which I think is clinically relevant and also for the large public. So first of all, methylation reactions are fundamental to our life, but they don't only pertain to epigenetics. And on the other side, epigenetics is not only about methylation. We have several classes of epigenetic marks, and I had an opportunity to work on 3 of the major classes of epigenetic marks. So we have non-coding RNAs, which are called non-coding because they don't produce proteins. We know that usually we have learned in the biology classes that the mRNA, the messenger RNA, is a molecule that that is a messenger for the production of proteins in our body. But these non-coding RNAs don't produce protein. Then there are histone modifications, which means histones are the protein that pack the DNA inside our cells and coil the DNA so that the DNA is very compact when it's closed and genes are off. And then when it's open, so histones and the DNA are open, the chromatin is open, then the gene is turned on. And this histone modification can be methylation, but also acetylation, phosphorylation, ubiquitinylation. So there are so many other marks. And then there is DNA methylation, so which is the methylation of the DNA itself. So not the histones, but the DNA, and specifically the cytosines, one of the 4 nucleotides of the DNA, within the context of so-called CG nucleotide, which is so the dinucleotide of C and G's sequentially together. Mm-hmm. And so to answer your questions, yes, methylation is one of the many epigenetic modifications, and it can be either on the DNA methylation, or on the histones. And but we can measure with much more accuracy the DNA methylation on our DNA, so the methylation on our DNA methylation, and that's why DNA methylation is one of the most used tools to measure epigenetic marks, especially in the context of lifestyle interventions.